you're here. I cannot activate the screen sharing for my presentation.
Should work now.
Ah, yeah. Okay, perfect. So let's see. So can everyone see the presentation?
Yes, perfect.
Okay.
Thank you.
Yeah. So my name is Philipp Hoenle. However, you can call me just Phil, like everyone does here. And I'm a new postdoc here at the Institute of Entomology in Patrick Clear's Ant Lab. I'm here since November, so not that new anymore. So most of you know me already. Yeah, I will talk today a little bit about my research that I've done in the Ecuadorian Choco. And this is mostly research from my PhD that I did at the University of Darmstadt under the supervision of Dr. Nico Blütgen. And yeah, as you already might have guessed, it's going to be a lot talk about ants and their ecology. And yeah, I'm glad to present here, even though I got Corona. So, but there's already quite some tradition for people to present, even though they have Corona. So let's see how it goes. Okay, now first to give you a little overview of what I'm going to present to you today. First, we're going to look at the ant community reassembly dynamics and land use legacies. Then I'm going to show you a chapter about the interaction of recovery and stratification. And finally, I will give you a short primer on my favorite ant group, which are the army ants. And we'll talk a little bit about army ant ecology and how it might matter in the context of regeneration and recovery. Now, you already saw here the background picture. It gives you quite a nice idea of how important ants are. So this is an Ectatoma tuberculatum worker drinking from an extra floral nectar, the sugar water that the plant produced. So this is from an Inga plant in Ecuador. And these plants are quite common as well as the ant species. So the ants obviously have some very important function for the plants so that the plants really give them sugar water for free, essentially. Of course, they expect also some kind of work from the ant that the ant does for the plant. So in this case, it's often protection from herbivores, but ants are also really important in other ecosystem functions, and actually quite a lot. Well, let's talk a little bit about the ecology of ants in general and why we as ecologists should really care about ants. Well, first of all, they can not only be friendly with the plants, but they can also be very important herbivores. This is particularly true in the neotropics, where we have the leaf-cutting ants as a very abundant and dominant group. So the leaf-cutting ants really eat huge amounts of plant matter, or to be more specific, they actually cut the leaves and feed a fungus and then eat that fungus. But they are also involved in a lot of more different ecosystem functions. They are important predators. They're involved in seed dispersal. They do soil bioturbation, you name it, ants do it. Just as an example for predation, I have here on the top picture, you see Pachycondyla impessa, which is more of a large-bodied single predator. And on the bottom, you see army ants, so more of a group predator in ants. And in this case, they were raiding a wasp nest. So you see a wasp pupa being carried by the army ants. So if we want to understand the ecosystem as a whole, in particular tropical ecosystems, it's really worth knowing what the ants are doing because they're so interconnected with everything else. So this is the first part of my talk. It has a title, Rapid Ant Community Reassembly, Recovery Dynamics and Land Use Legacy. This is the most important part of my PhD, and it was recently published in Ecological Applications. The background picture already gives you kind of an idea what it's all about. So you see one of the plots where my research. In the foreground, there's, so this used to be an old cacao plantation, and you can already see like a moldy cacao fruit in the foreground. So this is how a cacao plantation looks like after it has naturally recovered. Now, I always emphasize the region in which I do my research, the Choco region in Ecuador. This is because this is a really important tropical place. So most research in Ecuador in tropical ecosystem has been conducted in the Amazon, which is on the east side of the Andes. But the Choco rainforest is actually just this tiny little tip on the northwest of Ecuador, where it connects via Colombia to Panama to the more Mesoamerican forest system. So overall, this region is much more similar to Central American forests and to the Amazon, despite the Amazon being much more closer, geographically speaking. It's also a very highly threatened region. So we only have roughly 2% of the natural undisturbed forest left. And a huge part of it have transformed. Where I did my research, it was mostly transformed into small-scale farming, so into cacao plantation, cow pastures, and also, unfortunately, quite a lot of oil palm. But luckily, there's also a little bit of hope left for the Choco. So NGOs like the Fundación Choco, which were our close research partners during my PhD project. They own several reserves throughout Ecuador, and they are really committed in protecting what is left, but also in having ideas how to expand the conservation areas. And yeah, they are really thinking about the long-term future as well. Now, this particular reserve where I was is called the Rio Canande Reserve. It's named after the river that flows through there. And you see on the right-hand side below, you see a ferry crossing the Rio Canande, and this is almost daily picture. So actually the ferry and the whole connection to this conservation area is controlled by the logging company. And we see frequently trucks driving by with freshly chopped down rainforest. Now I also want to mention that I was essentially the first PhD student in a larger project, which is called the Reassembly Project, which is mainly about the recovery of interaction networks. And this was done it's like a main project from my professor Nico Plütgen, and it's just about starting right now. So it started like end of last year. So basically when I finished my PhD, but I had the honor of being the first PhD student to be involved in selecting the plots and so on. Now it's of course, for such a threatened area, it's a very important question of how long the forest will need to recover, if there's hope left, or if we need to like accept that there's only small rainforest fragments and anything that has been chopped down will never recover ever, so we don't even need to care about these areas anymore. Or if we can successfully integrate former pastures or cacao plantation into the forest and hope that it will regrow in a timely manner. And this is what my research focuses on. So now let's take a look how we can study this recovery. So luckily, Choco already a long time ago thought really in the long terms, and their conservation method works not only by buying already existing forest fragments, but also buying off small-scale farms in the vicinity of these areas and just letting them recover naturally. So over the years, Choco accumulated more and more of these farms. So we actually have really nice information on when these areas were left to recover naturally. and we can stitch these together to create a fake timeline. So like a space for time replacement is called a Chrono Sequence, to nicely study the recovery trajectory. So I have 61 plots in total for it. We also have year 0, which is basically currently ongoing cacao or pasture plantation. And as a sort of end goal, we also have old growth forest plots to compare these trajectories. And we can nicely distinguish between 2 legacies, so we can follow pastures and cacao separately in their recovery trajectory and see if there are differences between them. Now, I was, of course, interested in the ants. So I used 3 different collection methods to characterize the ant fauna on each of these plots. These are just standard morphological functional traits that we can assess from the species level. And then we can sort of have an idea how the functional aspects of the recovery work as well. Now, let's talk a little bit about the theory. What would we expect? What's happening during recovery? There's this nice paper by Moreno Mateos et al. Kind of summarized what we would expect. So, of course, it's highly depending on the community property that we look at, which kind of or how fast we would expect recovery. So, for instance, we would expect that species diversity would be faster in recovery than species composition, followed by interaction networks and so on. So this is a nice framework that I actually wanted to test with my real data. Now, as I mentioned earlier, I also had the kind of the functional aspects in mind. So I expected the functional recovery to be somewhere in between the species diversity and the species composition. Now let's apply our framework to what we have in the Choco. So as you can see here, we have 3 different recovery trajectories for each of the community properties, as well as for our 2 legacies. So now we can also get more information by looking at 2 different components of recovery, which are called resistance and resilience, which are mathematically speaking independent components of a recovery trajectory. So the resistance describes how much a parameter or the community property drops when we implement the disturbance, while the resilience describes basically the slope, so how fast it recovers. And this was my main research question. Now here we can see one of my pasture plots on the left side. So this is really typical of the agriculture there. We see it's not very intensive, it's more an extensive type of agriculture. A lot of trees are left standing for shadow, and it's actually also really close to the remnant forest, as you can see in the background of this pasture. Now on the right-hand side, we see a pasture that has recovered after 20 years. And you already see it looks kind of nice. So in the tropics, everything goes kind of fast. But how does it look like with the ants, of course? During my research, I found roughly 285 ant species, many of which have not been known to Ecuador, which are undescribed species, because I was one of the first researchers to do ant research in this rather remote area, which I'm kind of happy about. Now, I will not dive into nerdy details of the ants, but focus more on the ecology. So I just wanted to mention, I also described some ant species with a lot of help that I got from great researchers all over the world. But this will not be the focus today, so we'll skip this part. And yeah, let's look at species richness, a sort of measurement of species diversity. Surprisingly, we see that species richness doesn't really change across the Chrono Sequence. So we already have a really high richness levels in cacao and pastures. And we also see no differences in the in the legacy outcomes. So not much is happening here. Let's look at the traits. So this is an overall similarity index to the trait composition that we have in the primary forest of every plot. And we can see that the traits do show a recovery, though it's a quite fast recovery. So roughly 6 years for the cacao and roughly 9 years for the pasture. So there are definitely effects of the prior land use on the functional assembly. But these effects are rather small overall. Now let's look at the community composition. And this I find the most interesting. So we have really completely different ant communities in the agricultural areas, which are at the year 0, basically. And we see a kind of fast species turnover. And it takes roughly 21 years until the cacao legacy has completely recovered. So we cannot distinguish it anymore in their composition from old growth forests. And for the pasture, it takes a little bit longer. So we're talking about roughly 29 years until it's fully recovered. So there are some relatively strong legacy effects here, with the cacao having an overall advantage. Now let's look at the component of resistance and resilience that I mentioned earlier. And we can see that the differences that we saw in the time it takes for each of the community properties to recover is mainly driven through differences in resistance. While the resilience, so the slope, we cannot really distinguish well between each of the properties. And as expected, composition took the longest to recover, while it's a bit more muddy water between traits and richness. It depends what kind of measurements we're looking at, which one is faster and which one is not, but they're definitely much faster than the composition. So to summarize this, we saw that ant recovery is fast, roughly 3 decades. We saw that the cacao was doing a little bit better. This could be because of that the structure is already really similar to a forest, or more similar to a forest. If we have like little cacao trees, it's already kind of forest-like, whereas the pasture is really a different environment and it takes longer to recover. We saw that there is a lot of species turnover going on during the recovery, but surprisingly, it has relatively little effect on the overall traits. So we see a high functional redundancy of ants in this particular environment. It doesn't mean that this is everywhere like that. And you also have to consider that since all my plots are really close to the forest themselves, this of course reduces any effects that could be through habitat isolation and so on. And also the land use history overall is quite young. So we're talking about probably for most recovering areas, they have not been longer in use than 50 years. So this gives us quite a lot of hope for the recovery in the future. Okay. Now let's come to the second part of my talk, which focuses on sort of a new idea, which is, or not necessarily new idea. It focuses on the role of stratification in recovery. This is not yet published, but the chapter is pretty much finished. So it's currently just wandering from journal to journal. But of course, if you have any feedback for that, I'm thankful. Okay. Now, as you might know, especially in tropical forest, there's a lot of vertical stratification going on in the arthropod assemblages. So you have completely different fauna in the trees than on the ground, for instance. And this stratification is not only in the species themselves, but it's also different lineages, and they have really different morphology and presumably ecosystem function. So vertical stratification seems to be kind of important to consider, especially for ants, which have roughly like 50% of the species live in the trees, the other 50% on the ground. So considering this high stratification, the obvious question that comes to mind when we think about recovery time is, how do these both things work together? Do we expect that every kind of stratum reacts the same to recovery times, or do we see really distinct strata doing their own thing, let's say, and are kind of hard to predict from one stratum to another? And so I want to know, if I look, for example, at a single trait mean from a functional trait, how it reacts to both recovery and stratification. Now I collected with 3 different methods, which covers 3 different strata. So I had the winklers for kind of the leaf litter fauna, I had ground sampling for the ground ant fauna, and I had the tree trunk sampling for the tree trunks on every plot. Now, if we would, let's just take as an example, let's say body size, and we would see that the body size is stratified. So this is according to our stratification hypothesis A. We would expect that, for example, the smallest ants are in the leaf litter and the biggest ants are in the trees. And if it doesn't, if this functional trait is not shaped by recovery at all, we would just see these straight lines, like in the example A. If a trait gets shaped by both stratification and recovery, but recovery has the same effects on all strata, we would just see something like shown here. So for example, the ants get smaller across all strata equally. However, the more interesting and complex way would be sort of a statistical interaction between both the stratum and the recovery. And in this example, if we think of body size, so now here, the ants in the tree suddenly get bigger, but while the ants in the leaf litter, they don't change at all in their body size. It could be, there are many different possibilities. So it could be also that, so I have drawn 2 scenarios in C and D. So in C, we see that the strata actually become more different during recovery. So while on D, we see that they converge together. So overall functional similarity between the strata increases with recovery time. So my idea was to look at which of these hypotheses describes best the functional recovery of ants. First off, I just wanted to give you a quick look into the community composition. So this is just species identity, so it has nothing to do with traits yet. You can nicely see the differentiation between the strata. So we have a different community composition depending in which strata we look like. So T on the tree trunks, HS for the ground, and the W is more for the leaf litter fauna. And they are sort of separated, though there's obviously also a lot of overlap there. We can also see in the community composition our recovery trajectories. So on the left-hand side, we see like the currently ongoing agricultural areas, while on the right-hand side, we see more of the old growth areas. So in the 13 traits that I measured, I found that 11 of them were influenced by stratification in some way. So to make it more easier for you, I just have this overall figure. And I took the ground ant fauna as sort of a base template. And if something changed, if we went up to the tree trunks, then I colored it. And if something went down towards the leaf litter, I colored it. So you can see, for example, if we go from the ground to the tree ant community, we see that they get larger in body size. So the reverse length is marked here with colors. The eyes get bigger. the clypeus get bigger. This is sort of what we would actually expect from more arboreal ants. We also see differences in the phylogeny. So I made like the little genus level phylogenies for each of them based on previous published data by Economo et al. And now let's look at each of the strata, what happens if we take recovery into account. So you can go from each stratum and to the right-hand side, and you see colored in which traits are changing in each stratum. And overall, I found quite a lot of traits were shaped by the interaction hypothesis. So they were different from between the strata, to be exact, in 8 of them. Surprisingly, phylogenetic diversity was stable across recovery time, so I didn't draw another phylogeny there. Now, at the beginning, I told you about these two types of interactions that could happen. One was describing the convergence and the other the divergence. So here I have 2 traits. Actually, the majority of traits converged, 5 out of 8, where we have an interaction. So we can see the clypeus length, which is sort of an indication for ants that feed on sugar resources more. We can see that the clypeus overall gets smaller in across all strata, but not equally. So at the beginning, we have a much more stratification effect than at the end. And similar thing is happening with the color lightness. So we can see at the beginning the leaf litter is really out there. So has in color lightness, has high color lightness, so lighter ants. And over time, the other 2 strata kind of follow them. And at the end, they're not distinguishable anymore in the community weighted mean in this aspect. I also had some that showed the exact opposite trend, so the convergence. So for instance, or actually these are the only 2 traits that showed that, which are the Weber's length and the escape length. So the Weber's length is sort of an indication for body size, and the escape length is involved in the ant's perception, let's say, so the antenna and so on. And we can see that the strata become over time much more distinguished in their functional composition. So to conclude this chapter, we see that most traits are shaped by the interaction of recovery and stratification, which makes the functional reassembly kind of hard to predict. So we have to think about each stratum more or less separately in terms of their recovery. And ecologists should definitely keep that in mind when they, so whatever we see happening on the ground doesn't necessarily mean that we see it across the canopy as well. Okay, now let's come to my final chapter. I wanted to give you a quick primer on my favorite ant group, which are the army ants, and why they are really interesting organisms to look at. And I think every ecologist should have kind of heard about them. Now, army ants are really keystone predators in tropical forest ecosystems. So if you've been to the tropics, you probably experienced them yourself. The highest generic richness is in the neotropics. And they have a really distinct lifestyle with huge colonies, very interesting queen morphology. So I show you one picture of a queen below here. And they don't, in contrast to other ants, they don't develop wings and fly away, but actually the colony has to split up to sort of reproduce. They have a really nomadic lifestyle. So they move their nest. In most species, they move almost every day, except for certain life stage intervals. And fascinatingly, we can have roughly up to 20 species in one forest habitat living at the same time, which is surprising because you would expect they make each other a lot of competition and war going on. But it's actually not what's happening. Now, another thing I want to mention about army ants is that they're not just important in the perspective of the predators, but their interactions go much further than that. So this image shows you Echiton burchelli, which is one of the most common army ants in neotropical forests. And you see them during emigration. So they carry, in this case, a queen larvae from one place to their new nest, which they do almost every night. And if you look closer, you will see that there's an intruder in there. That's not an army ant. And it's this tiny little beetle sitting on the larvae, which is in this case called Cephaloplectus mus. And this is just one of roughly 300 known associate species that are living together and somehow associated just with this one species of army ants. And this goes much further than just little beetles. And there's even birds that are the famous ant birds that are specialized on following and living together just with the army ants. So yeah, you can imagine they have really a central role in the ecosystem. Now let's focus on an aspect of predation. We can distinguish two types of army ants. First off, we have the swarm raiding army ants. This is sort of what we intuitively know and hear in the news media about mostly. So they have these huge swarm fronts of ants that march through the forest and try to eat everything they can get. And here's an image from Daniel Kronauer of some typical prey items that he recovered during one of those swarm raids. And we see it's a diverse array of arthropod species of centipedes, cockroaches, beetles, but also a lot of other ants. And the other type, which is much more common than the swarm raiders, are the column raiders. So they don't form a distinct swarm front, but rather they have like just little streets going out. And they usually search for other ant nests and raid these ant nests. So we have a much more restricted prey type. In this case, it's a prey from Echiton hamatum. And we see a lot of leafcutter ant pupae in there. We see some Phydola in there. We see some Neoponera. So a quite diverse set of ants. But most notably, almost exclusively ant brood. And this is typical for a lot of army ant species. Now, they are especially interesting because we can use them as model organisms to study predation networks in the neotropics. I think they are really well suited for that because we have no or almost no intragroup predation. So one army ant will not raid another army ant. In fact, if they meet in the field, they will usually try to avoid each other. This makes also the ecological network between predator and prey much more simple because there's almost no intragroup predation going on. Then because of their biology, so they catch their food and then bring it to their nest where the larvae eat the food. We actually don't need to do any like gut matter barcoding or something to get an idea what they eat because we can just steal it from them on the way when they bring it back from their raid to the nest. And the ants will not, as far as we know, they don't snack beforehand on anything. So we get a pretty accurate view of the typical diet if we do a good quantitative sampling. And this is what I did during my master thesis in Costa Rica in 2017. And basically all we did was just walk in the forest of La Selva in Costa Rica. And every time we found an army ant raid, we would collect 10 prey items randomly, or like the first 10 that we saw. And then we moved on and looked for the next ones. So over time, in 3 months, we accumulated 238 raids from 11 army ant species. And we found that the whole army ant, or almost all army ants are really specialized on ant species. And we found basically a fourth of the complete ant fauna just as diet in the army ants weights. So you can see nicely on the right-hand side, a bipartite network between the army ants on the left side and the prey species on the right side. And you can see by the thickness of the bars of how common the army ants were and how common their prey items were. And each line connects basically each army ant species to their prey. And we can see there's relatively little or surprisingly little overlap in the diet, even though almost all of them eat exclusively ants. So prey items that made up no non-ant species are also marked in this graph. It's kind of a tiny bar, and it's actually only or almost only found in Echiton borchelli, which is in this case the only swarm raiding species. But even though it's such a famous swarm raiding species, they still get kind of a lot of prey from ants as well. So basically, we see this kind of high specialization on different ant species, and probably this is the main mechanisms that allows them to have so many army ant species living side by side. So they don't overlap in their diet, so there is not much competition going on. Now, of course, the coolest thing would be to kind of get predation. So if you think in the context of forest recovery, it would be really cool to have predation networks along the chrono sequence and then analyze that. And then was actually one of the main ideas of my PhD when I first proposed it, but it was a way too ambitious project. So until now, most of the data is not analyzed yet. But I can already show you that I collected and found a lot of the same species in Ecuador and in Costa Rica. So here just a few examples. For example, in the left corner, you see Echiton hamatum, and these little white thingies that is carrying, these are Phaidole pupae. And then on the ground, you see in the left down corner, you see Lapidus spinoides, which is a swarm raiding ant that actually loves to eat earthworms. So it's not everything ants, of course. And this is how far we've analyzed our Ecuador network so far. This was mainly done by a master's student in Darmstadt, Christoph Merkel, who did his master thesis last year on a comparison between the Costa Rica Army Ant Network and the Ecuador Army Ant Network. But we, as I said, we are still kind of analyzing the prey. So this is a project that will take a little bit longer. Now, unfortunately, I don't have the spatial resolution to do a good chrono sequence because the army ants, the sampling was just not sufficient enough. And the army ants are not common enough to get it really plot-wise. But maybe, or very likely in future, we will try to pull it down to like recovering networks and old growth networks, for instance, and try to get an idea what happens to the predation networks overall. For instance, if the specialization increases or decreases. One thing that was interesting in the comparison between Costa Rica and Ecuador networks is that we have more swarm raiding species. So I marked them with blue here. So we have two extra species of swarm raiders in there. And we will this time also identify non-army and non-ant prey down to species level. But yeah, this is still in work. I just wanted to give you a quick show of what we've done so far. And I hope we can make a nice research paper with that in the end. Yeah, I just wanted to show you some faces of people that were involved in my study. And as I mentioned, I'm mostly thankful for Choco to allow us to do our research, and the Reassembly Project for the good ideas, mainly Nico Blütgen. But yeah, there were countless other people involved in my ant research. To conclude, overall, I showed you that the tropical forest can recover fast, at least from the perspective of the ants. I actually expect that other insect groups will show similar recovery trajectories. I think they should be correlated with the ants given how interconnected they are. We learned that stratification matters for functional recovery, and maybe ecologists should always keep that in mind. And yeah, the Choco has many more hidden gems awaiting discovery, so many more nice ant species, but also other species, of course. And I hope it will be preserved for the future, and there will be a lot more future studies in this very promising area. Now, since we have some time left here, I just wanted to give you a very quick summary of what I'm doing now here. So as I said at the beginning, I'm a postdoc in Peter Klime's Ant Lab, and I will soon go to Papua New Guinea to help with the experiments there. And the idea is also to get more idea how succession affects the functional and phylogenetic diversity, especially of arboreal ants. And we plan to have really big manipulative experiment with small bamboo nests. And we kind of want to move around ants from a high elevation place to lower elevation place to simulate the effects of climate change and so on. And also I'm reanalyzing some older data. So one of them is almost done and is from a former chapter of Nicola Plowman, and where we surprisingly find that ants in secondary forests have a higher functional diversity than in the primary forest. But yeah, you will maybe soon read that. Okay, thank you for your attention. And are there any questions?
Okay, if there is, maybe people need some time to think about the questions. Thank you very much again for the really nice talk. And I was wondering about the study where you was talking about the body parts and the phylogeny which changes along the vertical stratification. Maybe I missed, how was the phylogeny incorporated in the traits, or how?
So, let's go back. So basically, I did the phylogeny and the traits independently of each other. And in the analysis, what we did was just calculate phylogenetic diversity itself, and try to see if it changes across recovery or across stratification. And it only changed across stratification. So on the trees, they actually had a higher phylogenetic diversity, but this didn't change at all across recovery time.
Okay. So the phylogeny was not incorporated in the traits as such. The traits were like the colors, they were analyzed separately.
Yes, yes. So it's quite likely that a lot of the traits have a phylogenetic signal, but we kind of didn't explore this much further.
And do you think it might be a problem, or you will think about that for future, or you think it's not necessary?
I think for this case, it's not so necessary, since we, as I said, we don't see much happening in the phylogeny actually. So whatever signal we get out of the traits, we can already assume that it's something that's sort of happening independent of the phylogeny, or at least on the genus level phylogeny. It would be best if we had species tips, but this was just too much work to do, so we just had a really rough genus level phylogeny. So there might be some nuances that we're missing here, so we have to be kind of aware of that.
Okay, thank you very much. Because
Thank
we
you.
are just thinking about something similar, but only in the primary forest for the beetles, and we actually didn't get to that. We are just still collecting the data, but students
Okay,
will be
nice.
doing that soon. So I
Yeah.
was, maybe we will come to you with the questions, how to deal with it.
Okay, yeah, why not?
Yeah, thank you. Are there some questions? You can just speak loudly.
I thought that when I was in Panama and met some Ceton army ants, that they predate mostly on wasps. I mean, the true wasp Vesperi. Was it true as well in Ecuador? Or the wasps were not so common there? Or the ants were more specialized to other ants?
Yeah, yeah, no, you made absolutely a correct observation. So you see this quite frequently, and a lot of army ant species love wasp nests. And you even it's kind of often obvious when they do that, because you might have like a lot of wasp nests around your hut sitting there, and then one day the army ants come and they just eat everything up, and the wasps are super helpless and they can't do anything. And so we have wasps in our predation data. I also showed at the very beginning a picture of a Butrelli eating a wasp. The wasp is here. But if we really quantify it, it actually doesn't make much, or it's really tiny fraction of the whole food. So this is a typical example of something that you might see as very often in the forest itself, but you usually don't notice when they raid a lot of ant nests around you. So you get kind of a, let's say, distorted view of how much this actually contributes to army ant prey. And there's just not enough wasp nests around to feed the army ants as a whole. So they take it as sort of a free snack, but in the grander scheme of things, it only makes up a small fraction of their total prey.
Nice, thank you.
Hi, Phil.
Hey.
Very nice talk. Thank you.
Thank you.
I have just follow-up questions of Katka question about the phylogeny. When you have this data, you actually can just have a look if the soil living ants are not more phylogenetically diverse, but maybe more ancestral, because you have nice data to do it. You have genus level data, so you can maybe look at it. Maybe you already looked at it. So
yeah. No, thank you. Yeah, I can do that. Actually, I expect them to be more ancestral. So my initial expectation was that the ants on the ground or underground should be much more ancestral and have a higher phylogenetic diversity. But I think the reason why I found higher phylogenetic diversity in the trees was that it's kind of hard to compare my collection methods with each other in this sense, because maybe just there's some bias that I got more ants on the trees in general and less in the ground. So it didn't reflect like really well in the overall phylogenetic diversity.
Okay.
This could be a reason why that's not the case. Yeah.
Yeah, well, that's just another question for more people doing similar data and similar phylogenetic analysis. But I would expect that, but I didn't really saw the results like that in the literature. So I was just curious about it.
Yeah. Thank you.
But maybe follow-up question, and how you sample the soil living ants? Did you dig up the soil pits or?
So I would love to do like really like underground pitfalls or something like that. But for my research, in this case, it was important that we can do like all the collecting on one plot in one day. So there was no time for placing pitfall traps. So the more the lower strata was done with winklers, and with the winklers we always took like parts of the topsoil actually in the pasture we cut out like grass turf and placed that in the Winkler traps at the end. So we don't really get like the real underground ant fauna, but sort of an in-between mix of like more leaf litter ants and a little bit more deeper living ants.
Okay,
So
that's more
it's mostly
yeah,
mostly leaf litter ants.
Okay, yeah, that's maybe more clear now. Yeah, thanks.
Any more questions, please? I don't see anyone to activate now. Oh, seems, Phil, that your talk was really nice and everything is clear to everyone. So you will go to PNG soon?
Yes.
Okay,
so
I hope.
Next month is the plan.
Okay, so thank you very much once again for the overview of your work before and shortly about now, and good luck in the field.
Yeah, thank you very much.
Thank you and bye.
Goodbye, everyone.